So let me talk a little bit about the research center. And our Research Center for Ornamental Fishes is the research center's name was founded in 2014 by myself and a, um, a fish biologist here in Peru. We're located in Iquitos, Peru, which is in the very northeast corner of Peru in the Peruvian Amazon. Here is Iquitos. Iquitos is a city of about 700,000 people. The only way you can get to Iquitos is either take the Amazon River from the mouth of the Amazon uh, down to Iquitos or upstream to Iquitos, or you fly into it. So it's an island all to itself. It's surrounded by three major rivers. We have the Amazon River to the north, the Rio Nanay on the west side, and on the east side is the Itaya River. So river life plays a very important role in all the livelihoods of the people here in Iquitos. If you go to um, well, the research center itself sits at about 7.5 kilometers away from the Iquitos Airport, um, on the only road that leads out of Iquitos, and it stops 50 kilometers south of Iquitos at Rio Tigre. And if you go to and you can Amazon will take you right to the research center. So I guess we've made it big because Google has accepted us as one of their own. So this is a drone photo of the overall research center complex. Um, we have the um, office here. In the back side is a small instrument lab where we have microscopes and basic research equipment. The large building here is our quarantine house, where we call this the fish house. We use it to quarantine fish before we put them into the aquarium. And also this is where our researchers work if they're working on fish in aquaria and doing observations. This back building here is our dormitory complex with a kitchen facility, dining room facility, and a laundry facility. We have one of the few private dryers in all of Iquitos. They don't use dryers here. They usually put things out on the line, but during the time when it's more than 100% humidity, things tend to turn black because they get mold all over them. So I don't like to wear moldy shirts. My wife doesn't like me to wear moldy shirts either. Here is the aquarium of the Amazon. Um, this was our most recent building that we built. We'll come back to what we do with the aquarium itself. Here we have research ponds uh, where we conduct experiments on sustainable aquaculture, trying to breed some of the ornamental fishes of this area. And in the back, we have our reservoir. Water flows naturally and percolates into this pond because the water table is so high. So normally, unless it's an extreme dry period, uh, we use the water from all of our work at the research center. So as Tim said, you know, the research center is here to help preserve the biodiversity of the Amazon. We look at it in a couple of different approaches. We look at conservation, sustainability issues. We provide hands-on education for the school kids. We support research by graduate students and undergraduate students at the National University of Peru, and from students from the States, as well as we host international researchers that come and do research here in Iquitos. And we have infrastructure. We established an education research hub here at the research center itself. One of the things we do is we go out to the villages, the fishing villages. The aquarium industry is fairly large here, but we go out to the fishing villages and teach them how to look for diseases and things that are going to impact their catch. We treat them how to undertake basic treatment. We try to get away from use of antibiotics and other chemicals. And so we're teaching them how to use natural products that they have within their own environment so they don't have a great impact on the aquatic environment themselves. Um, we teach them how to undertake good transport of their fish so they get the top dollar for their catch. One of the things that happens though is they always sell through intermediaries, and the intermediaries will buy fish at a very low price. Let's say Otocinclus affinis, the common Otocinclus, the intermediary will buy 1,000 Otocinclus for $1. He then turns around and sells those same Otocinclus to the exporters for 50 cents each. So that intermediary is making a big profit off these fishermen. And so what we're trying to do is teach them how to transport their fish themselves so they can get a better market price for their own fish. And here with Carlos, who is our resident manager here, our general manager here, is dissecting fish to look at, what's the fish all about? Looking at their gills to look at gill parasites, their intestinal tract, because most of these people have never seen the inside of a fish unless they've cut it open to eat it. And then how to, keep their fish alive once they catch them, so they're at, better quality when they sell these fish to the exporters. We undertake a major environmental education outreach here at the Research Center during COVID. Well, our mascot is Nalda and Eduardo. Nalda is means strength in Spanish. So this is Nalda, our mascot, and our Eduardo, who she captured. So what my wife has done with Nalda and Eduardo is she has written two books, two little workbooks that we have the little kids work through. Um, and what Nalda and Eduardo do is they teach certain environmental questions and environmental processes and what we can do to help preserve the environment. We're not hitting them over the head with it. It's just they're a fun story to help to see what Nalda and Eduardo, what kind of troubles they can get into and how they overcome them. So during COVID, we actually took the books to the schools. We give away all the books. We don't make any money off the books. We lose money, in fact, because we're giving all the books away. Um, Now that we're out of COVID, we bring kids to the research center and they work on little projects together, do little demonstrations. Um, Here is a school that we brought in, School for the Deaf here in Iquitos, and we did presentations for them. Again, they all get the book. And here's a picture of a classroom with their Nalda books in front of our statue of Nalda at the public aquarium that we have here. This was the opening to the public aquarium showing all of our staff. And one of our board members who is right here. Unfortunately, not all the board members could show up. Tim decided he was too good to show up for our opening since he is one of our board members as well. I should disclose that, so I'm not sure if you all know that. So this is our public forum. As you walk in, we have what's called the tunnel tank. The little kids like to walk through that. They pretend like they're underwater. It gives them, a little bit of a different view than just looking at an aquarium up, from the glass up front. This is our stingray tank. Stingrays are one of the fish that are really affected by overcollection. And traditionally, when people in the Amazon see a stingray in a stream, they will kill it because the alternative is for them to get hit by a stingray. And because it's so humid here, that injury takes a long while to heal. So they just as soon kill the stingray than have to worry about, stepping on another one. Um, so the room is surrounded by tanks, all different types of fish, all the common fishes that we see in the aquarium industry here. You ask people how many fish they know in the Amazon, and they will name you 5 fish because those are the fish that they eat. They're not concerned really about any other fish, and most people have never, even that live here in Iquitos, have never seen these fish that they have in their own backyard. Of course, everybody knows the infamous red belly piranha. We put a fake alligator skull in there to show that the piranha uh, you know, ate the alligator huel in there. His family was eaten by piranhas. They thought it would be the best thing, but no one can take a joke, I guess. Just more tanks, electric eels, mata-mata turtles. So not only fish, but different aquatic life. Leaf fish. You can see the leaf fish right there in the middle. We do have one marine exhibit. Even the kids here in Iquitos know who Nemo and Dory are. And what we do is we make the connection between what's happening in freshwater will eventually affect what happens in the ocean. My favorite tank, of course, is the flooded forest tank, which is a tank full of wild Peruvian discus and rummy nose tetras that we both exist together in the Rio Nanay. We know that though the discus aren't native to Rio Nanay, so it kind of is an exotic species here. But despite that, no one here knows that. This happened in the 70s, and most of the people who were around them then are no longer here. But so most people think they are native to the Rio Nanay when in fact they are not native. One of the rooms I thought we should have, the exhibits, is a nocturnal room. Most people here, if not all people here, will never go into the water at night because they are very super and they drown them. So they do not know any of the fish for sure that come out at night. So most of these fish are knife fish, some of the bigger catfish, and we have exhibits to show the fish that they have. And one thing we do every year is that we have a Christmas party for the kids in the village. We get upwards to sometimes 400 kids show up. For many of these kids, this is the only Christmas celebration they have. They don't teach them about Papa Noel or Santa Claus because how can you say to a poor kid, well, Santa Claus didn't bring you a gift this year. So we have a big celebration for them a few days before Christmas, and every kid who comes gets a gift. And for again, some of these kids, these are the only gifts that they'll get for Christmas. So it's one of we try to be good neighbors to the people there in our little village that the research center is found in. The people who work at the research center listen to people. What research do we do? This picture is of Dr. Eric Thomas, who does work on Corydoras poison glands. And so he comes to the research center. He'll be here in a couple of weeks to continue his work on poison glands and Corydoras. We are working with red pencilfish. The red pencil fish are found in where it says starting location. It's an area about a 20-hour boat ride away from Iquitos. It has a very small, restricted geographic area that it lives in. Um, and the problem is that this place is being overrun with massive environmental degradation. In fact, there are stories here that this Peruvian village, which is Alvarenga, was attacked by giant yellow-eyed aliens in 2023. And there was, if you go to YouTube, there's over an hour and a half special on YouTube about this alien attack. And they had an expedition there of these UFO experts to go and talk to these people. What it ended up being is that the gold miners attacked these people at night, dressed up in all these horrific costumes, and started cutting the people's faces because the gold miners wanted to chase away the villagers so they can have free range of this area to have unlimited gold mining. That's one of the problems that they have here in Peru is that there is illegal gold mining all over the place and the government does nothing about it because the gold miners pay off the government officials. And so it's it's like the wild, wild west here when it comes to gold mining. One of the things we also do at the research center, we are the only receiving center in Iquitos to receive any fish that are confiscated in the exportation process. These are two fish that at certain years, they're illegal to export. This is the tigrinus catfish and the Jurensis catfish, which is related to the tigrinus. If they're exported, you have to have special permits. If the exporter does not have special permits to export these fish, when they're allowed to export them, they are confiscated by the customs agents, and we receive all these fish. Last year, we received a shipment of 20 boxes of fish of primarily Corydoras catfish because in order to export, you must have an exportation facility, and this person did not have an exportation facility. And so we received all those fish here at the research center. We put some in the aquarium. Others we just keep in our fish house, and we're the custodians of the fish. We keep them until they die, which some fish right away. Other fish we've had for 5, 6 years that are still going strong here at the research center. We do, as Tim mentioned, we do primary surveys. We do surveys using seines, surveys using hand nets, and we also do surveys for some of the more cryptic species of using environmental DNA to determine what's found in the water. And some of the fish we found, red lizardfish are one of the popular fish in the US. They do occur naturally here in a stream here in this area. We find red fish, we find brown fish, and we find black fish. So everybody thinks the red lizardfish, the loricarid, is a different species. If you look at the L number, it actually has a different L number, but in fact, it's the same species as the normal lizardfish that you have, the loricarid that you have. I forgot the name of the catfish now. I'm getting old. But they are undescribed species of catfish. Even though they're two different colors here, they're actually the same species when you look at them genetically. Uh, it may be male, female, or maybe just different morphs of the same species. Plecos, a lot of plecos here that we find that are undescribed. Here are two pictures of the same pleco species. So now let's talk about our research. So threats to the Amazon, the threats that we have here in the aquatic ecosystem is really the same threats you have all over the world. Of course, global climate change is the big one that affects everything in the environment. Introduction of exotic species. Exotic species does massive destruction all over the world. And those are the two that I'm going to talk about in the rest of this seminar. But we do have habitat loss and destruction, and of course, overexploitation of species stocks. There are fishes here in the Iquitos region that have been devastated by overcollection for the aquarium trade, one of which is the chameleon loricarid. Uh, it, it is one that um, takes 2 or 3 days now to collect the same amount of fish that it used to take the fishermen an hour to catch. Because once they discovered where it was found, they completely devastated the fish there. They wiped out the local populations, and the local populations are just barely coming back to life now. But it is a major problem here in this area. So let's talk about global climate change. Of course, we know that the temperatures in the the argument can be made, is it natural or human induced? As a scientist, I think there's plenty of evidence to suggest that it's human induced. But if we look at what's happening on the land, uh, the temperature increase on land is even going at a much greater rate. It's not just this 0.7 centigrade degrees, but it's much higher than that we see in the ocean or in the aquatic ecosystems, because the aquatic ecosystems water is a good temperature buffer, whereas land, especially over the hours, much less over long-term years. So in freshwater, global climate change is even greater because fish can't disperse from one lake to another. It's not like you have, mammals that can move, especially some of these large mammals that can move to areas that are more conducive to living. A fish, once it's in a stream, once it's in a lake, even a riverine system, it cannot go more northern or southern to get away from those temperature extremes. So they're going to be cruelly impacted by these temperature changes. Here in the Amazon, over the past 10, 15 years, we've had major um, droughts that have occurred, droughts at the time when there should be high water. So the rains did not come one year when they were supposed to come, and they never came that year. And that devastated the aquatic environment here. So, catastrophic droughts, the worst droughts in 100 years, 40 years. Then following year, we have these dramatic floods. So not only do we have the droughts going on, well, we have a complete changeover in the seasonality of some of the rains. And this is really what's happening with the global climate change here in the Amazon, is that you're going to get these massive droughts and massive floods occurring over a cycle that where it's just going to change the environment completely. Despite the fact that, we know what's happening in global climate change in in the system to climate change in the ocean.
Anthony,
But
sorry,
if
I'm
we
just
look
interrupting.
at what's happened in fresh water,
Anthony, would you
yep,
mind just going back just the start of that blue screen about you were talking about the marine systems? You broke up again slightly. I didn't quite catch what you said.
okay,
Thank you.
okay, sorry about that.
No, no, it's not your
So
fault.
from 1983 to 2020, it's been over 150,000 papers written on the impacts of global climate change on marine systems. And if you look at that same time span for freshwater systems, there's less than 1,000 papers. So most science really is concerned with what's happening in the ocean. Very little science is concerned with what's going to happen in freshwater systems. So we know the greatest impact will be that as temperature increases, the water loses the capacity to hold oxygen. And so that will affect them right away. And we see this in a lot of fish when the temperature increases. Some fish have the ability to come to the surface to gulp air. Carp will do this. The labyrinth fishes will do this since they have a labyrinth organ to extract the oxygen. But other fish don't have as good a capacity to extract atmospheric oxygen like many other fish do. And in fact, you get massive die-offs at certain times of the year when the oxygen is stripped from the water. This is a massive die-off of tilapia that are an invasive species in a saline lake in Southern California where I grew up. And it happens every year that millions of these fish die, and they are such prolific breeders that they're producing so many offspring that a million fish every year doesn't have much of an impact on the population. So let's talk about what's happening, what potentially could happen in some of our killifish. Of course, since it's a killifish group, you all know Notobranchus guntheri. They're found in the seasonal pools and streams in parts of Africa. They go through a diapause period, so the eggs have the ability to dry out. They grow very fast, 6 to 8 weeks. They can reach sexual maturity and start breeding. We know they're brightly colored because you want to have a mate, as attractive mate, because your lifespan is so short in these pools that if you wait too long, your genes are then not being passed off to the next generation. And so you become an evolutionary dead end for this species as an individual. So in the normal temperature that you find these fish, the air temperature when the ponds and the water dries up is 25 degrees Celsius. So what one of my students and I did is we conducted an experiment looking at the hatching success in Notobranchus guntheri when the peat that the eggs were laid in were incubated at 25 degrees Celsius, 30 degrees Celsius, and 35 degrees Celsius. Yes, 35, massive temperature, but she wanted to go up to 35 just to see what would happen. And so this is what we found. When incubation temperatures were at 25 degrees Celsius, the normal temperatures, with the 50 eggs that we had in the peat, 42 of them, so 84% hatched in the first treatment. In the second treatment, 90% of them hatched. You bump up the temperature to 30 degrees Celsius. We see that the hatching success is dramatically decreased. 10%, and about 9% in between those two treatments. When we go up to 35 degrees Celsius, none of them survived. The eggs never hatched, and in fact, they had problems with them that we looked at under the microscope. And one of the things that was happening at these higher temperatures is that in the peat that that these fish were spawned in and incubated in these higher temperatures, these nematodes all of a sudden sprouted out and were consuming the eggs. At 25 degrees Celsius, we did not see the nematodes um, in that soil when we looked down under the microscope. And here are just normal hatchlings of the fry in the uh, 25 degrees Celsius temperatures. The fry either had these large curvatures in their spines. Again, I know. And in others, they had this lordosis at the back of near the caudal peduncle area. And that just, they did not swim normally with those problems. So as we get to higher temperatures, these fish are exposed to much higher temperatures in the sand, and that will affect the hatching success and the viability of these fry once they do hatch. So let's now move on to exotic species and how they can affect killifish. Peru began introducing exotic fish in the waterways here when they introduced rainbow trout in some of the high altitude lakes to provide a better sport fishing for the people. But what happened was the rainbow trout that were introduced into these lakes started consuming some of the native fishes. And in fact, one of the native fishes that was a food fish for the people in the higher latitude area or higher altitude areas became extinct. So they wiped out a whole species of fish just to provide more entertainment for the sport fishers. The three fish that we work with here are guppies, which were brought here for mosquito control. Uh, blue gourami, which are not eaten here in Iquitos, but are stocked in some of the aquaculture ponds to help keep down the algae populations and the plant populations that invade the aquaculture ponds. Now, guppies is what we're going to talk about. Guppies play an important role here because people collect bags maybe and for less than $1 each, and they are sold to the exporters to feed their fish that they have at the export facilities. And there's a close-up. You can see all the baby guppies. These are the wild type guppies. You do find some nice delta tail guppies occasionally show up in some of these streams and some of the areas that you find guppies, but mostly it's the wild type guppies. And here is one of the wild type guppies. You have two different populations here, a more red colored population and a more blue colored population. The population that we are working with were the red population. So we know that guppies have high fecundity. They give rise to a lot of young. There's been studies to show that the mean average young that a female produces is 23, and she can reproduce every 28 days on average. Takes her about 2 to 3 months to mature. So one female can theoretically give rise to over 1,000 offspring per year. If you look at her children, her grandchildren, her great grandchildren, and so on. So a pregnant female can wreak havoc on an area, especially since they can store sperm for up to 3 successive broods. So if we look at guppy distribution all over Iquitos, you can see every place that there's a G, we found guppies. We also Peat Lake. It receives its water only from water that percolates through the soil and leaf litter. So it's a very low pH lake, dark stained lake. So we find guppies there. We find guppies in some of the streams around the lake as well. So what impacts do the guppies have on the environment here in Peru, particularly killifish? In areas where we find guppies, we don't find any killifish. And in areas that we find killifish, we don't find guppies. So we're looking primarily at the small streams that one particular killifish lives in, which is Ribulus rectocaudatus, which is shown in this aquarium glacier photo. And this is a typical stream that we find rectocaudatus in. Again, if there's guppies, there'll be no killifish. If there's killifish, no guppies. So what's happening with that interaction between those two? So we conducted 2 experiments at the university back in California. We took these microcosms, these 435 liter containers, and we stocked them with either guppies, killifish, or a combination of guppy and killifish. And we monitored them over 90 days. So in tank 1, we had 25 guppies and 25 killifish. Tank 2, again, 25 and 25. In tank 3, 50 guppies. And in tank 4, 50 killifish. After the 90 days, we emptied the tanks and counted the number of fish that we had in the tanks. And this is what we found. In tank 1 that had both, we had 102 guppies and only 15 killifish. In tank 2, we had 225 guppies and 23 killifish. In tank 3, 187 guppies. And in tank 4, 47 killifish. So, in tank 1 and tank 2, there was something happening here. More than likely what happened is the killifish that we did not see or did not find, more than likely jumped out of these tubs. Killifish are great jumpers, as many of you know, if not all of you know, and there was only about 6-7 centimeters from the water's edge to the top of the tank. So it could have been that they jumped out of these tubs. So what we did secondarily is that we did direct observations on the fish. So we brought them into the lab. We had 2 different tank systems. They were 150 liter tank 1. In one systems of tanks, we had 10 adult guppies and 10 ribulus in there. And the other set, we had 20 ribulus only. And we made direct observations every day for 30 days. Now, these little brown objects on the bottom are actually Indian almond leaves. We have Indian almond leaves here in Iquitos. The trees were brought in during the early 1900s during the rubber boom.
Hopefully not. Anthony, sorry, could you go back? Just talk
And the
about
tanks that we had,
Anthony.
yes.
Can you just go back? You're just talking about the Indian rubber the almond leaves being introduced in the 1920s, I think, or whenever, and then we lost you for a moment. I think we may have lost you again.
They brought in a lot of Chinese workers and a lot of Southeast Asian workers, and they brought in with them plants to make it more like home. So one of the plants they brought in were Indian almond leaves or Indian almond trees, catapa trees. And so we have them all over Iquitos. In fact, we have them on the research center that we put the leaves on the bottom. Um, just it was we find these leaves in the environment in many of these streams, so we wanted to replicate what the streams were. So when we had both guppies and, and killifish in the same tank, you know, they're surface feeders and surface fish, but they would swim all over a tank like guppies do, and the riverless would stay more in the center of the tanks. Now, people say, oh, well, this is typically what happens. You know, the guppies act as dither fish, or the killifish act as dither fish for the guppies, whatever. And so, you know, a normal thing that would happen is that, you know, fish would distribute by where they're normally found in the environment. But if we look at when Rectocaudatus was by itself, this is what we saw. The fish stay under the leaves and only eat the food that would find its way under the leaves. So what is really happening? Well, if you look at what is happening in the environment, when you go out and collect Rivulus rectocaudatus in these streams around here, they only are under the leaf litter. You don't see them in the water column. And you can sit there for hours observing the streams and you never see one of these fish. And so their normal habits are to live under the leaf litter. And so they only go out in the open when there's not enough food for them. So the guppies are out competing them for food. So they have to be more, they throw caution to the wind. They have to, you know, go out and be more open in the environment, which makes them more prone to predators. So it's not their normal behavior. And in fact, once the experiment was over, we kept the tanks going just because we were going to do some other experiments a little bit later on. And what we found is that in the Rectus caudatus tanks only, we had fry emerge. In the tanks with guppies and the killifish together, no fry from these may have been happening was that the guppies were feeding off the eggs, not feeding the eggs, but were eating the eggs from these killifish. Now, we never saw a direct observation. We didn't, you know, sacrificing the guppies to look at their gut contents. Or they may be eating but you know, it just seemed a little bit more than we thought was going to happen to have these fry emerge from the riblets only tanks. So what can killifish tell us about the environment? Well, they're a fantastic bioindicator species in these tropical regions. You can get a good idea of the water quality, the habitat quality, the food web stability, whether, you know, there's adequate food that meets the needs of all the individuals, all the different species in that environment, as well as they're a good bioindicator for what's happening in global climate change in the local aquatic systems in the tropic regions that you find killifish. And with that, I say stop share. I know as an American, I speak really fast. I hope I didn't speak throwing rocks at the screen, anything.
Anthony, that was fantastic. This poor man's been awake for 28 hours, and to do that, present that, before I've learned a lot there. That was wonderful. Thank you. Come on, people, let's have some interesting questions.
Thank you.
Either in the chat box or just unmute. I think it won't be too chaotic because we've only got like 13 people on here.
So I've got a question. With regard to the observations of temperature effects on Guantheri eggs,
Yep.
I had two questions / points. One is they're not spawning in peat in nature; they're
Yes,
spawning
that's
in
true.
dirt,
Yep.
right? So
Yep.
a few inches down is a lot colder. And secondly, I just did a quick, I hate to say it, I used, Google AI. What are the temperature ranges for Guantheri habitats from the names I could remember, like Mafia Island
Yep.
and
Mhm.
stuff like that, and it gets as high as 32.
Oh, okay.
So yeah, and 25 seemed to be the bottom end. So colder than
Yep.
25 wasn't going to be normal, but as high as 32 should have been fine. So anyway, it's an interesting experiment, but I don't,
Yep.
It's a little squishy
Oh yeah,
in
yeah.
my
No,
view. It's not
these
reflecting
were
nature.
yeah, these were donated by a local breeder, and we used 25 because that, for one thing, that's what he was incubating his fish at for generations. So his fish were selected for those temperatures. And so that's, one of the reasons why we chose that temperature as well.
Mm-hmm.
Sure, it doesn't reflect what it may be happening in their native ranges, but, may give some type of indication
Right.
of what the
And
species
I
can
think the
handle once
the
it's, been under certain conditions for many, many generations.
mhm. The other thing was the nematodes. I'm questioning the peat moss there. Was it sterilized? Was it, or he's in Iquitos, Peru, or wherever. Where's he getting his peat moss?
Well,
Garden center.
this was actually in the States. So this study was conducted at
Okay.
the,
Yeah. Yeah. I don't know. It's an
so,
interesting experiment, but I think
yeah,
it could be
yeah, yeah.
could be done again. Yeah.
Oh, it could be done under better, there's no doubt, under better conditions. Yeah. This was, you know, this was an
Yeah.
undergraduate student doing the research. And so it wasn't, it was to teach her how to do research, to get some good background information, but,
Okay. Yeah. I'm not denying that global warming
yeah,
is affecting freshwater habitats. It's
yeah,
glaringly obvious it is. So
yeah,
it's not just marine, but the marine gets all the headlines because of
yeah, yeah,
weather.
yeah, yeah,
Yeah.
yeah.
Quick question,
Okay, thank you.
Anthony. Does the eDNA, you're telling you the species, is it giving you a population indicator as well? Is it a population indicator?
It it can be used based on the concentration of E and DNA you get. They can use it to get some sort of population numbers.
Okay.
Not in terms of there's 100 individuals out there. It will tell you whether or not it's a good, viable, population number, or is it rare? But
Right.
one of the problems we have here in the Amazon is all the water we're testing is an acidic water, and DNA degrades very rapidly under acidic conditions.
Okay.
So, even if a species is found in high population numbers, if we're trying to use it to say, yes, there's high population, we may get very little DNA, which indicates they're low in population, but in fact, because it's degrading so rapidly. So we don't use it here to indicate population numbers at all. It's presence or absence. But again,
Okay.
even if it's absent, doesn't necessarily mean they're not there. That's why we do the eDNA surveys with manual surveys. We always use seines and hand nets, at the same time.
I was going to say, which do you do first? Is there
Yeah.
do you do eDNA first and then say, we've got this killie in this
Yes, of
puddle,
course. Yeah.
and then you start
Yeah.
having a proper look
Yeah.
for it?
Yeah. Yeah.
Got you. Thank you. Stephen.
The 4 tanks that you had, where you had guppies in one, rivulus in another, and then you had guppies and the rivulus. You reckon the rivulus jumped out, which I can well believe.
Yeah.
Could that experiment be done again with perhaps some netting over the top to give you
Oh, yeah.
a more?
Yeah.
Yeah.
Yes. The problem we had with the Rectocaudatus is that I had to import all these fish. Even the guppies were wild caught guppies from Peru. And to import fish, for me, you need 10 boxes of fish to make it worthwhile, because I once imported 1 box of fish, and that importation cost me $400 at the time, where an importation of 10 boxes, which is 100 kilograms, cost only $1,000. And so as a professor on a tight budget, I couldn't import as often as I wanted to.
Right.
Just a quick question. Have you noticed in your surveys, even over the time that you've been surveying, any dramatic changes in distributions?
We see a dramatic changes in numbers of fish. Not necessarily distributions per se, but there's a lot of habitat degradation going on here. A lot of new settlements springing up, where 10 years ago this was a pristine area, where there was the streams are nice and crystal clear. These settlements have open sewage systems, so all that sewage is going straight into the waterways without going through a treatment facility. So it's becoming worse and worse here.
The UK then.
Yeah.
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 Any more for any more? I think William's just far too dozy this evening.
Yeah.
Out in the sunshine.
Looks like Peter has one.
It does. Hello, Peter. Peter, could you unmute?
William, they said you're looking a bit dozy there. I said, have you got cans of Guinness there or something?
No.
Okay.
I didn't get your question about eDNA. Is that are you looking in dirt in the lake to find DNA and then
We're looking in the
figuring
waterways. We're in the water itself.
okay.
The one killie we're working with is not an annual killie. Rectocaudatus is not an annual killie, so it lays its eggs in the stream. So there are some, of course, there's some annual killies here, but the one we were working with before was Rectocaudatus.
And it's the cells you're detecting. Are they
No, you're detecting
cells?
You're detecting parts of the DNA, which is they use DNA barcodes, which are small portions of DNA that are unique to each species.
And
But are they
Sorry.
They're floating in the water. Sorry, Tim.
Yeah, the DNA is
yeah.
just floating in the water. Yes. Yeah.
Okay.
Peter has just asked the question: do you liaise with Project Piaba in Brazil?
We do not liaise. We're quite different than Project Piaba. They're more well established. They've been around for everybody. Every, you know,
I'm going to ask you to repeat that if you can hear me,
20,
Anthony.
30 years. They get all
Sorry,
the money.
Anthony.
We look. Oh, yeah. So, they've been around forever. They have a big stable of supporters because they've been around forever. Everybody knows Project Piaba. 5 people know about ARCOF. They look at the aquarium industry a little bit different than we do. They look at the aquarium industry as it's sustainable to collect these fish because they're only really working with cardinal tetras. That's it. Even though you look at Project Piaba and their emblem now is a discus, they don't work with discus. They only work with cardinal tetras. That is what they say is sustainable. And tetras are sustainable to a certain point because they have a very high reproductive output. I'll use some scientific jargon. They're called R species, which means they're short-lived individuals, highly reproductive, per reproductive output. We look at it differently. We're primarily working with, for sustainability, some of the cichlids, especially Apistogramma. And these can be over-collected very quickly. And so we're going out to these villages, and something I didn't talk about, but we're going out to these villages and we're teaching them how to sustainably breed these fish. And so they have a steady income all year long, for one thing, because they can only fish during the dry season, which is right now. They can't fish during the rainy season. But if they're breeding these fish in small ponds, 2 by 2 meters with 1 meter deep, they can have a steady income all year long for these fish, which means potentially they could fish less. We're not telling them that's what they should do because that's their livelihood, but at least they have an income during the winter time, so they don't go out and do destructive agriculture or cutting down timber, things of that sort. And so I just got back from Singapore last week, where I gave a talk at the World Aquaculture Conference on the work we're doing, because we've done in our ponds here, we've actually showed that this concept can be done. And in fact, in our aquaculture ponds with our Apistogramma, we're covering the aquaculture ponds with mosquito net, and we're not even feeding hundreds of offspring from these Apistogramma just from them eating the microorganisms that occur naturally with decomposition of all the plant material that's in the water. And so that's, we're going this summer to a 30-hour boat ride away to teach them the concepts of sustainable fisheries. So they look at, sustainability in catching. We look at sustainability more so in terms of there is overcollection. Let's try to rectify that problem and maintaining an income for these people.
May I just ask which Apistogrammas? Ed would like to know which Apistogrammas you
we've done Apistogramma Jurensis, Apistogramma pandurini, the sunset Apistogramma alto tapayo. So all the Apistogrammas that we find around here, we show that they're easily cultivated in these ponds and it's non-intensive, that we're not feeding them. And that was important to us because these fishermen, they don't have money to spend on fish food. I wish we had $1 million grant where we could provide them fish food, but we're providing the training, and we're giving them a system where it's very little outflow from their pocketbook, and still getting an income from them.
Fantastic.
Why do you think marine gets all the attention
Well,
with the
primarily
research
because,
for?
it's our seafood base. Most of the fish that we eat, most of the seafood that we eat comes from the ocean. And it's much more highly visible, the ocean and what we get out of it than freshwater systems.
How
And
can we
it's,
change that story around? Is it possible, or is it just the way it is?
Look at our Earth. It's covered 70% with salt water. Less than 1% is, less than 1/10% is fresh water that we can see. Most of it's in the ice caps. So, it's such a minor constituent of the environment that, people take it for granted.
Yeah.
And the coral bleaching events are so spectacular. They make good news, don't they? It's good, good copy.
Yep.
Yeah.
Yeah. Yep.
Okay. Any more? 'Cause it's 5 I'm very conscious that Anthony needs to get to bed. So
No, I'm not leaving you to bed. It's only what we're 6 hours behind you. It's still in the afternoon. If I go to sleep now, I'll be up by 2 o'clock.
Okay. May I ask a quick question then? Are you detecting any
Doesn't do me any
mercury?
good.
Are you detecting any mercury in your?
Yes, yes, Rio Nanai, which is one of, for this area, that's getting heavily hit by illegal gold mining. And there was just one study that came out probably 6 months ago on the larger fish, showing that there is mercury levels in the fish.
So it's in the food chain now for the.
It's in the food chain, yes, yeah. And we tried to sound the alarm, but I think sometimes they look at us as alarmists because when I first started working on the gourami problem here, some of these streams have billions of gourami. And the head of the environmental protection region here, who's a fish biologist, denied there were gourami in the water. And one of the slides that I showed when I gave a presentation to the government was an area right by his house showing a seine full of gourami. At that point, he had to say, "Oh, I guess we do have gouramis." And the government was allowing them to take gourami from the environment and taking them out all over the Iquitos Amazon to these aquaculture farms to feed Arapima.
Okay.
To have a natural food source. But all these farms flood into the rivers here during the flood season. And they wanted to take gourami and transplant them to the native environments of the Arapima to give the Arapima, a better chance of surviving. Luckily, because we said no, gourami can't be used in the Arapima farms anymore, and the government decided it'd be better not to do it. But again, what happens here, there's no enforcement. So I'm sure it's happening. There's no enforcement for the gold mining.
It's very sad, isn't it?
It's sad. I love this place, but it's sad what's happening. And sometimes you can, they stop listening to you when you say the same things over and over again. And that's why we really decided to do the education route for the kids too, because that's how you change the society is through the kids.
Fantastic, yes.
Can I ask one last question? Sorry, it was about I'm hearing some more positive things from Brazil with the new government about a reduction in deforestation and other thing. Early days. is that having any impact on Peru?
Yes and no. They just elected a new president, Keiko Fujimura. She's been president in the past. Her father was president, and they tend to run the government like a dictatorship. And her father he laid down some environmental laws that were strictly enforced. And the first time Keiko was president, she also did the same thing. She was booted out of the presidency and spent 10 years in prison for corruption. But they still love her here, and so they just elected her, and some people are hoping that she'll have that same dictatorial rule, and which will help the environmental processes.
I
But
don't know whether to say fingers crossed or not to that, but I mean, at
yeah,
least
yeah,
the
yeah.
environment, it sounds
Yeah.
slightly positive. No,
Yeah.
thank you.
Yeah, human rights, she doesn't believe in human rights, but last time she did believe in some environmental rights.
Well, we've got lots of humans.
Yeah, exactly.
Okay. 9 o'clock. Any more, or should we call it a night? I've still got to put my fish to bed.
Yeah, I've got to see to mine as well now.
Okay, so I think we'll wrap up. Anthony, thank you again. That was fantastic. Absolutely.
Thank you all.
Brilliant
I
talk.
appreciate the invitation.
It's been our pleasure. And
Yeah.
let's say that was a special one. Thank you ever so
I
much.
learned a lot there.
Yeah,
Thank you.
Very good. Thank you.
good.
Yeah,
Right.
thank
Great
you,
evening,
Tony.
everybody.
Thank you.
Thank
Take
you.
care, everyone. That's
Bye.
Thank you, Anthony.
Thanks.
Bye-bye.
Bye-bye.
Bye.
Thank you. Bye.
Bye. Bye.